Assalamu alaikum. So let's discuss how to generate long-lasting recombinant human growth hormone. We have discussed this strategy earlier in other classes of our health biotechnology, that addition of extra amino acid to N terminus or the C terminus can exponentially increase the half-life of that recombinant drug, biotechnology protein, therapeutic protein, or any other biotechnology protein of importance. So this is what was done by the scientists when they generated long-lasting recombinant human growth hormones. What they did, they actually added extracellular domain of human growth hormone receptor. So they fused the receptor portion with the human growth hormone. And this fusion actually took place via a linker peptide. And that linker peptide was 20 amino acid long. And it actually consisted of 4 repeats of a block. And that block consisted of 5 amino acid. Out of them, 4 were glycine and 1 was serine. So 4 glycine, 1 serine unit was repeated 4 times. That made a total of 5 multiplied by 4 is equal to 20. So 20 amino acid long linker peptide was used to fuse the human growth hormone, which was recombinant in nature, with a small portion of the receptor for that human growth hormone. And keep in mind, the human growth hormone receptor's extracellular domain portion was added there. So when this construct was tested in rat, a single injection resulted in promoted growth for 10 days. And that was very significant difference, because for the regular recombinant growth hormones without these fusion partners, daily injections were needed for those rats. But with this single infusion, with this single procedure, it drastically increased the number of days needed for the new injection, 10 days. The similar results were found in macaca. Macaca are actually the monkeys. You can see here the figure for the picture for the macaca is also shown here. Look at the graph, which indicates how the growth hormone and the modified growth hormone levels were observed in the rat blood following the subcutaneous injection of the relevant recombinant human growth hormone. You can see the red line indicates the regular one, and the blue line indicates the modified growth hormone. And you can see that the levels remained quite high for longer as compared to the other one. You can see that till 60 hours, the levels were kind of higher uh, than the compared version of the regular growth hormone. Okay, G. So you can see in this figure, you can see in this figure that the growth hormone was derivatized by coupling it to a portion of the human growth hormone receptor, which we have just discussed. So the first A portion is indicating our growth hormone, the peptide linker, and the receptor fragment. So that can actually dimerize. So when it will dimerize, that will stabilize our recombinant growth hormone in vivo, causing it approximately 300 times slow plasma clearance from the kidneys than the free human growth hormone. And when it is needed, when it gets bound to the growth hormone receptor, it conveys the very similar response which the non-recombinant or the non-modified version does. So the recent development in human growth hormone, if we see how they are behaving, how they worked in the clinical trials, so you will be happy to know that the fusion human growth hormone without the linker was also constructed and tested, and it yielded 1.7 times higher levels of the human growth hormone. So when the linker was missed, but the receptor portion remained there. Therefore, all subsequent studies, which included human trials, they are now using the fusion protein without the linker protein, and only the receptor protein is present over there. And it is very good or beneficial because it does not require addition manipulation steps. So ultimately, when the steps, the subsequent purification or downstream steps are reduced, then it becomes more cost effective. And it is particularly important in resource limited setups such as Pakistan, which is a developing nation. So currently, I'm referring to the GLIC 5th edition 2017 stuff, where they said that currently it is in phase 2 clinical trial. But this actually gives room for our today's assignment, which is that we have to check the status of the clinical trial for this human growth hormone through different registries. You can check them on clinicaltrials.gov, which is US maintained database, which listed all the ongoing clinical trials as well as the completed clinical trials worldwide. Then the second registry is International Clinical Trial Registry Platform, ICTRP. It also provides access to the global clinical trial registries. I have also given the opening phase which you are going to see when you will go and search for these sites. Okay, G. The assignment is that you have to look for where the clinical trials for human growth hormones stand in 2025, whether they have been completed, whether they have been approved, or whether they are still under investigations. Now we have to look at another very, very interesting and concerning use of human growth hormone, and that is doping with it. Actually, the human growth hormone is readily taken by some of the athletes worldwide for improving their athletic performance. It is also used by males and females for the skin rejuvenation purposes, and the bodybuilders also use it. And let me tell you a very important and interesting incident related to it, and that is back in 2010. Doping with human growth hormone is not new. It has been in use since 1970s. But later, when it was identified, then it was banned by International Olympics Committee. And the interesting thing was that the synthetic human growth hormone cannot be detected via urine analysis. So for those who were using the synthetic one, they remain got undetected. They were never identified until the development of another test which can correctly and accurately identify the recombinant or synthetic human growth hormones back in 2010. And the first athlete who was identified using this human growth hormone, he doped with it, was the British rugby player Terry Newton. There is another aspect to it which is traumatic, that he got so depressed when he was caught using recombinant human growth hormone that he actually had suicide in September 2010. So I have added this case specifically for you all to know that one should be very careful in using human growth hormone, recombinant human growth hormone, only for situations where medical interventions are needed. If you are using it for athletic performance increase or any other purposes, then it is better not to use it altogether. You can also see that it is present in different packings of kg, 5 kg, etc. for the bodybuilders, and one such example is the GH force which I have shown there. So now I am stopping this talk here so that we can discuss the next thing later.